name is Daniel Sherbenou, the University of Colorado Chutes Medical Campus in the Division of Hematology, and I'm here to discuss our abstract at the ASH meeting in 2023. The title of our abstract was that high ex vivo response rates to BCMA CD3 bispecific antibody Elranatamab in patients relapsed after anti-CD38 and anti-BCMA targeted immunotherapies. In our lab, we have a platform for measurement of ex vivo drug responses, including to immunotherapies. And recently, we have been using this to measure the responses of patient sample myeloma cells to bispecific antibodies. The way this works is that we obtain bone marrow aspirate from patients with multiple myeloma, and we treat these samples for 48 hours in culture. These are cultures that include all the immune cells that are present in patients' bone marrow that help fight multiple myeloma. And then at the end of the 48-hour period, we use a machine called a flow cytometer to measure the decrease in the viable or the live multiple myeloma cells. And we use this as a measurement to see how well this drug works in patient sample. So this graph on the right here shows that the patient's T cells are required for this class of medication to work. The way a bispecific antibody works, for example, Elranatamab under this study, it basically binds to a multiple myeloma cell through one arm and has another binding arm that binds to T cells. And by proximity, brings those T cells in and engages them to kill the multiple myeloma cells from that patient. This is a new class of therapy that's very promising in multiple myeloma, and we now have 3 agents that are approved. As you can see in the graph on the right, T cells are required for that process. When we remove T cells, there's really no appreciable killing. So killing is on the y-axis of this graph, and so a deeper response here, a lower number indicates killing of the multiple myeloma cells in the assay. So we've been using this assay with Elranatamab to ask the question of if it is an effective agent in patients that have had previous anti-BCMA CAR T cells, because the target is the same. They both go after this protein that's called BCMA on the surface of the multiple myeloma cells. As I mentioned, there are 3 drugs that are approved in this category. So teclistamab is the first, Elranatamab is the second, and then the third shown here is a drug called talquetamab. All of these agents work through the same general mechanism, with the exception that talquetamab binds to a different protein on multiple myeloma cells called GPRC5D. Our hypothesis for this abstract was that most patients who relapse after CAR T that also targets BCMA have been found to still express high levels of BCMA on the surface of their multiple myeloma cells, and thus would hopefully be good candidates for this kind of therapy that still targets BCMA. In our assay, we found that this drug Elranatamab, this bispecific antibody, very potently kills multiple myeloma cells from patient samples. This is an initial study where we made what's called a dose response curve, testing from very small concentrations to larger and larger concentrations of the drug. And you can see that with increasing amounts of drug, all the multiple myeloma cells are killed in the culture. And we basically are finding what we expect to find, that this type of drug is very, very potent. There's only a small amount, which we call a picomolar amount, that's required to induce complete killing. So we've been basically picked a concentration of 1 nanomolar that we expect to kill all the multiple myeloma cells from a patient's sample, and we use that single concentration to screen a larger number of patients in a kind of a high throughput fashion. So this is what's called a waterfall plot, where we're looking at patients who are newly diagnosed pre-BCMA, meaning they haven't had a therapy that targets BCMA yet, and then post-BCMA, which, as I mentioned, our hypothesis, we're hoping this drug still works for this category of patients. And you can see overall there's maybe some patients that are resistant even in the newly diagnosed setting, meaning they don't get to this dotted line at 80% viable, so 80% of the controls, so 80% of the cells are still alive. But in the post-BCMA group, we're seeing promising activity that, although a small number of patients at the time of our abstract, they're all responding to this relatively well. So with this initial information in hand, we can start to ask questions about, are there certain biomarkers of response? What that means is, can we predict by measuring certain features in the patient's multiple myeloma cells or in their T cells that will enable us to predict, is one patient going to respond better than another, and identify good candidates for this treatment. And so far, the left graph here shows that the target expression of BCMA, the levels of BCMA on the surface, which is graphed on the x-axis here, does not actually predict how well this drug is going to work. Basically, what we're finding is that most patients express BCMA on their multiple myeloma cells to a fairly high degree, meaning, and 10 here would mean like a 10-fold overexpression on the multiple myeloma cells compared to a background level. Even in patients that have a pretty low or almost undetectable level of BCMA on their surface, they're still responding fairly well. So this drug is able to induce responses despite low levels of the target on the multiple myeloma cells. And the two that here we show that are resistant to the drug, that don't have an adequate response, it appears that maybe the explanation in one, this 1802, could be that that patient had a very low number of T cells, or vice versa, just a lot of multiple myeloma cells, so that the T cells were very, very much outnumbered and not able to overcome the very high number of myeloma cells. And then one sample, we don't have a good explanation for why that sample didn't respond. The multiple myeloma cells expressed a lot of BCMA, and they also had a lot of, shown here, and then they also have high levels of T cells present as well. So we're working on getting more replicates to see if we can identify biomarkers going forward, but so far this looks promising for most patients. So one question we're further interested in is there an identical type of T cell or an identical or an optimal scenario for which the T cells should be utilized from the patient? So for example, if the patient is relapsing after CAR T, would the T cells still be effective in attacking multiple myeloma cells? And here we're finding actually that there is some indication in only one example so far, but we're looking at studying this more, that T cells from a healthy donor are more effective at killing the multiple myeloma cells than T cells from a patient. So this is something that we're interested in studying more. Now, we started studying in a couple more patients shown here, whether T cells that were obtained from the patient before they got CAR T cells and at the time when they relapsed after receiving CAR T cells, for example, 1 or 2 years later. And we're actually finding that in these patients so far, that the T cells from the relapse after CAR T are still as good at killing their multiple myeloma cells as they were before CAR T. This is encouraging because it shows that the T cell fitness or their functionality is still preserved in that post-CAR T relapse setting. There's another medication that I mentioned, Activism. This is also an agent, a bispecific antibody that targets BCMA, also FDA approved. We're finding that when we looked at comparing these in a small group of patients so far, it looks like their activity is highly correlated to each other, like you might expect. When Elranatamab works really well, Activism also works really well. And vice versa, when we identify resistance to one agent, it's also present in the other. This has only been done in 4 patients thus far, but so far they look like their responses to Activism on the y-axis and Elranatamab on the x-axis are highly correlated. So in conclusion, we're finding that MyDST is a useful tool to help identify biomarkers of how patients are going to respond best to this new class of agent. And we're interested in using this assay to hopefully one day guide treatment with this class of agent and other drugs for multiple myeloma, and also identify ways that we can optimize the T cell setting for which patients are treated.